Hello, my dear students. I'm Dr. Vaishali Bharambe. I've been teaching anatomy for the last 25 years and I love it. This lecture is a part of my histology lecture series. Today and for the next 3 lectures, we're going to discuss epithelium. Today is the first lecture of this series. Okay, so epithelium part 1. So we should understand that human body has got tissues. What is a tissue? Cells aggregate together and function in a collective manner to form tissues. So you can have, that means that cells are together in a particular region and they are functioning in a collective manner together, okay, for a purpose. For example, you have epithelial tissue, you have muscular tissue, you have connective tissue, and you have nervous tissue. These are the 4 types of tissues in our body. Today we are studying the epithelial tissue. So what do you understand by epithelium? Let's not look at the screen, let's think. What is epithelium? A collection of cells, yes, which are closely adherent to each other, okay? They form a sensible layer, coherent layer, okay? And they are either lining the body from outside or lining the body from inside. Does it work for you? Collection of cells in close opposition to each other. Can I have one cell here? Can I have one cell here in epithelium? No. They're all opposed to each other, forming a coherent layer, a sensible layer, covering external surface of the body or lining internal surface of the body. That is epithelium. So, what are its characteristics? The characteristics are important. All epithelia are avascular, closely opposed to each other, held together by cell junctions. Can I separate cells in epithelium? Normally not. They attach to each other with the help of cell junctions, resting on a basement membrane. You can use this image here to understand better. Having functional and morphological polarity. This sentence I have put here purposely. I want you to understand that if this is a cell, the apical region of the cell has a different function, lateral region is different, and the basal region has different functions. They are individually, they have polarity. Okay, so avascular, held to each other with the help of cell junctions, resting on a basement membrane with functional and morphological polarity. That is characteristic of any epithelium. Function, now don't bother to remember. Just use the mnemonic PASTER. Epithelium will protect, so protective. Epithelium will absorb, absorptive. Epithelium will secrete, secretory. Epithelium will transport material across it, so transportability. Excrete, E, it will excrete. And finally R, it will act as receptor. So let's take a look. Protective, absorptive, secretory, transport, excretion, reception. PASTER. 6 functions of epithelium. Very easy to remember and also easy to understand the epithelium if you know its functions. Development of epithelium. Why am I teaching you this? Because functioning of epithelium becomes easier to understand if you know its development. See, epithelium has developed from ectoderm as well as endoderm. That epithelium which is covering your body from outside, including your skin, your cornea, your conjunctiva, is all ectodermal. Some bits of this ectoderm, take a look here, dips down into the underlying mesoderm, finally forming gland. This is how your sweat glands are formed, your sebaceous glands are formed, your mammary gland is formed. This is how they are formed. So epithelium forms glands also. So deeper portion of that gland will be secretory and the top portion will be the duct. Similarly, endoderm lines the entire body cavity, oral cavity, your esophagus, stomach, everything. Okay, this also forms by invaginating and proliferation will result in formation of gastric gland, liver, pancreas. Okay, very few epithelia in the body are actually mesodermal, like lining of peritoneal cavity, etc. Just understand the basic concept. Let's move on. What have we covered up till now, students? We have done definition, we have done characteristics, we have done functions, and we have done development of epithelium. Let's now begin to classify it. So students, suppose the epithelium has got one layer or it has got multiple layers. You can classify the epithelium on the basis of this. So based on number of layers and based on the kind of cell you have in the top layer of epithelium, you are going to classify your epithelium. Single layered, simple epithelium. Multi-layered, stratified epithelium. Based on the shape of the cell, if the cell on the top is flat, you call it squamous epithelium. If the height and the width of the cell are the same, you call it cuboidal epithelium. If the height is more than the width, you call it columnar epithelium. Okay? So, but this is only the topmost cell. In an epithelium, what is the type of topmost cell? That will decide the epithelium. So, combine these together and let's classify the epithelium as simple and stratified, simple squamous, simple cuboidal, simple columnar. Sensible? And additional pseudostratified epithelium. I'll tell you what it is. Stratified again, stratified squamous, stratified cuboidal, stratified columnar, and transitional. Today we are going to discuss only the simple. Epithelium 2 is when we will be discussing stratified epithelium. The link for this is given in the description section. Feel free to see that as well. Simple epithelium. First, simple squamous epithelium. What do you mean by squamous? The word squamous means a scale. Okay, the fish has got scales. It's that word. So, an epithelium which is as flat like the scales of a fish, so that the nucleus is flattened, cytoplasm is hardly visible. That is a simple squamous epithelium. If you look at it from the top, it looks like mosaics. Mosaic tiles, it looks like mosaics from the top. So, thin layer, plate-like cells with flattened nucleus, seen from the top looks like a mosaic. Found at places where quick diffusion is needed. Can you have a nice thick layer for crossing oxygen from your lung into the blood vessels? No, oxygen won't cross. By that time you will be dead. So you have to have very thin epithelium for oxygen to easily cross it and be absorbed, right? So it is found at places where quick filtration is needed. Example, lining of alveoli, lining of blood vessels, mesothelium, and so on. Let's take a look at this image now. Can you see this is the alveolus of a lung? Can you see flattened cells lining it? Imagine oxygen entering inside, crossing this flattened cell to enter into the underlying blood vessel. As you can see, that blood vessel is also lined by simple squamous epithelium for allowing quick diffusion. Okay, that is simple squamous. Let's now talk about simple cuboidal. That means the epithelium will have cells, single layer, simple. Each cell will have the same height and width, cube. Okay. So where do you find it typically? Nuclei are rounded in the center. Typically, you will find this in your thyroid follicle. You can see it here. Okay. You can see how the follicles are individually lined by cuboidal cells. You may also find this over the covering of ovary, germinal epithelium, simple cuboidal epithelium. Or maybe ducts of glands here. This is a tubular gland. You can see how it's lined by cuboidal cells. Or proximal convoluted tubules are also cuboidal cells. So this is simple cuboidal epithelium. What about simple columnar? Common sense. Height of the cell will be more than the width of the cell. The nucleus is somewhat elongated and more basally placed. That is simple columnar epithelium. However, this epithelium shows a lot of modifications. Okay, so if it is simple columnar epithelium, plain and simple with no modification, you find it lining parts of stomach. It can have a modification in it called as a goblet cell. Okay, so what is a goblet cell? Take a look at this image here. You can see this cell, the apical region of which is full of mucinogen granules. Okay, so what happens is, during the staining, these granules are washed off. and what remains is the cell that looks like this goblet cell here, empty on the top. Okay, so simple columnar with goblet cells is this epithelium. This portion is the portion that is lost. Okay, right. Sometimes the top of the cell gets folded into millions of tiny folds which are called as microvilli. Okay, take a look at this image now. Can you see the apical region of the cell membrane folded with a core of cytoplasm? These are microvilli. They increase the surface area of the cell primarily for absorption. Okay. So, if these microvilli are straight and evenly placed, we call it a striated border. If they are clumped together like wet eyelashes, then you call it a brush border, typically found in gallbladder. Sometimes the apical region folds into long folds. Okay, such long folds are called cilia. Okay, so then you have simple columnar epithelium with cilia found in fallopian tube. Okay, can you see these cilia? So you must be thinking, ma'am, microvilli, cilia, same thing. They are folds of the apical region. How to differentiate? Are they really different? Yes, they are different. Microvilli are non-motile. Cilia are motile. This is a Viva question. Please prepare this well. Both have a covering of cell membrane with a core of cytoplasm. But microvilli have a core of microfilaments, whereas cilia have a core of microtubules. Very, very important. Do not forget this. Okay. Microvilli are thin, short. Cilia are thick, long, comparatively. Microvilli are many per cell. Cilia are fewer per cell in comparison. Microvilli perform the function of absorption by increasing surface area. What do cilia do? Cilia perform the function of motility. How? I'll show it to you by the end of the class. They are important for movement of material in the lumen of the organ they are lining. We'll discuss this later again. So what have we learnt up till now about simple epithelium? We've learnt there is simple squamous epithelium, cuboidal epithelium, columnar. We learnt that columnar comes with modifications such as columnar with goblet, columnar with microvilli, columnar with cilia. Then we talked about examples of each. Then we discussed differences between microvilli and cilia. So, we're done with simple epithelium but one, which is pseudostratified columnar epithelium. Pseudo? What is the meaning of pseudo? Pseudo means false. Pseudostratified. That means it's false stratified. It is something that looks stratified but is not stratified. Okay? So what is the meaning of this? It's such a complicated statement. Basically understand that all cells are resting on a basement membrane, but all the cells do not reach the lumen. Some are short, some are long. So the nuclei are variably placed. If you are looking through a microscope, you may not see the cell wall at all. So you see multiple layers of nuclei and that you assume it to be stratified, but it is actually not stratified. It is pseudostratified. Okay? So that is pseudostratified epithelium. All cells do not reach the lumen of the organ. They are often covered by cilia, which cilia are never found on stratified epithelium. So this epithelium is often covered by cilia. So pseudostratified epithelium can be without cilia, can be with cilia. Without cilia you find in male urethra. With cilia you find typically in your respiratory tract, lining trachea, bronchi. Okay, that is very typical. It may also have a few goblet cells. You can see goblet cells here. Okay. So that is pseudostratified ciliated columnar epithelium. Question is, how do these cilia function? Remember, I had told you I would teach you that. So look at this carefully now, okay? See how there is the arrow is representing some mucus in your lungs. The cilia will beat, pushing the mucus forward. Cilia will beat to push the mucus forwards. This is how mucus that is collected in your lungs gets pushed up and then you spit it out or swallow it, whatever. But it is no longer in your lungs. Question: what will happen if the cilia are absent? This material that is produced in your lungs stays in your lungs, causing consolidation, causing pneumonia, or even causing repeated sinusitis. This condition is called as Cartagener's syndrome or immotile cilia syndrome. This patient will have repeated lung infections, repeated sinusitis. If he is a male, he may also complain of infertility. Why? Why infertility? Because the tail of the sperm is actually the same microtubular structure. So, when you have immotile cilia syndrome, the sperms are also found to be immotile and therefore person is infertile. Right. So, what have we learnt about simple epithelium today? We learnt about simple squamous, cuboidal, columnar epithelium. We learnt about different modifications of columnar. We learnt about examples, differences between microvilli and cilia. And finally, we talked about pseudostratified epithelium. I've clearly given you how these come in exams. Some are just identification slides. Some are proper short note, like pseudostratified epithelium. Differences between microvilli and cilia are viva questions. Please be prepared for those as well. Students, there are two related lectures that I want you to attend. One is a lecture on glands. Epithelium modifies itself to form glands. I want you to attend that. That comes under Epithelium Part 3 lecture. And then, of course, stratified epithelia comes as Epithelium Part 2 lecture. Attend both. Links are given in the description section. Unless you study all these three lectures, you will not have a thorough knowledge of epithelium. If you liked this lecture, do like and subscribe to my channel. Share your thoughts in the comment section. I really want to know if my lectures are really helping you in your study of anatomy. Thank you, students. See you across the screen in some other video.